Hi guys, hope you are doing well. Today we will discuss in our second lecture in chapter 7, the second part of chapter 7 dealing with the different types of the spontaneous and induced mutations. And at the end, we'll briefly discuss some of the DNA repair systems. So, we will start by the spontaneous mutation. لازم نعرفوا guys first about the definition of mutagenesis. This term is divided into two parts. Muta means mutation and genesis means creation or birth. So mutagenesis means the creation of a mutation. To create a mutation, this process can occur either spontaneously or can be induced. يعني يا أما لحالها فيها تصير الميوتيشن يا أما لازم يكون في عنا an external factor بيساعدنا to induce the mutation. When we say a spontaneous mutation guys are naturally occurring mutations. So نحن ما دخلنا ما بنأثر فيهم ونحن ما بنحط extra agents or chemicals بيعملوا لنا mutation naturally occurring. However, the induced mutations, لا هون عم نحكي عن if the organisms or the cells are exposed to mutagens. Mutagens means physical or chemical agents. Example, if we are exposed to radiations, radiations represent physical mutagens, UV radiations or X-ray radiations, or if the cells are exposed to chemical mutagens such as the chemicals present in the cigarette smoke, some types of preservatives and food. Sometimes barbecuing will transform some non-toxic compound into carcinogen, يعني leading to mutation and then cancer. Some types of products present or chemicals present in cosmetics. So all these types of chemicals and physical radiations representing mutagens means induced mutation at the level of the DNA. So two types of mutations, guys. يا أما spontaneous mutation or induced mutation. Please keep in mind that the induced mutation occurs at a frequency higher than the spontaneous mutation. So رح نقسم هيدا باقي السلايدز guys ل two types. Spontaneous mutation أول شيء رح نحكي عنها, then we will move to the induced mutation. Let's start by spontaneous mutation, guys. تتذكروا in our previous lecture I defined the term point mutation. وقلنا point mutation example missense, nonsense, neutral, silent, transition, transversion. So in general, all types of point mutation occur spontaneously, which means you can change one base into another, you can substitute one base into another, or you can add one base or delete one base from the DNA sequence by spontaneous reactions. Where we can introduce spontaneous mutations. These mutations mainly occur at the level of the DNA replication. So this is the cell cycle, guys. During DNA replication, when you replicate the DNA, أكثر شيء ممكن يصير في عنا errors, mutation during the S phase of the cell cycle. But also you can find spontaneous mutation in the other stages of the cell cycle, such as the G1 and the G2 representing the growth phases. And during mitosis or meiosis اللي هن ده cell division. راح نبلش ناخد one example. اتفقنا انه قبل شوي انه during the cell cycle, during the S phase, during DNA replication, spontaneous mutations or errors can occur. During this process, guys, base pair substitution mutation, when you substitute or replace one base to another, one base pair أكيد to another. Here at this level, an error can be introduced. Why? Because the DNA polymerase is responsible in the enzyme responsible in the introduction of a new nucleotide complementary to the base present in the template DNA sequence. At this level, the DNA polymerase sometimes can introduce a mismatched base. Example: تخيلوا انتم بالتمبليت عندكم A، نحن بدنا نتوقع انه the DNA polymerase رح تشوف ال A and introduce a T facing the A, right? Complementary to A. But sometimes by error شافت ال A بس بدل ما تحط T حطت C. C is considered as a mismatched base to the A. So C represent an error introduced during DNA replication. هيدي one form. في عنا كمان another case, guys. How we can introduce errors during DNA replication. بدي تشوفوا معي please هيدي الفيجر, guys. كلنا بنعرف we have 4 different bases: thymine, guanine, cytosine, and adenine. These 4 different types of bases are present in a normal common form. The common forms, guys, are known by keto for thymine and guanine, and هون بتشوفوا عند السيتوسين والأدينين the amino form NH2 NH2. بينما بالجوانين and thymine we have the ketone C double bond O C double bond O. So the common normal forms present in our cells are keto or amino form. But also the 4 bases present in our cells can be present in a rare alternative form known by tautomer. شو معناتها tautomer, guys؟ إذا منبلش guys بالثايمين and guanine اللي هن بنسميهم keto form، شايفين C double bond O C double bond O اللي هن the keto form. If you convert the C double bond O, the keto form, into an enol form, اللي هي the COH double bond، شايفينها COH double bond representing the enol form. This enol form, guys, represent a rare form of the base and are known by tautomers. So when you convert the normal common form into the rare tautomer form, this process is known by a tautomeric shift. نفس الconcept is applied to the cytosine and adenine اللي هن the amino form. كمان we will change the amino form NH2 into an imino form اللي هي the NH double bond C. لاحظين, guys? So here you will change the form from amino form to imino form. It's the rare tautomer form. So this change from normal to imino is known by a tautomeric change. In the common normal forms, guys, نحن بنعرف we have the normal base pairing بنسميهم the Watson and the Crick base pairing. معناتها بنعرف T will base pair with A و C will base pair with G. هيدي if the bases are present in their normal form, the keto or the amino form. However, when you change the form to the alternative rare form, the tautomer form of the bases, now the bases will start to match with other types of bases. يعني for example, رح ناخد example. Let's consider we have a thymine, okay? This form of thymine, it's a common form. But if guanine is converted into the tautomeric form, the rare form, alternative rare form, tautomeric form, the rare form of the guanine now can base pair with thymine instead of cytosine. So لاحظتوا وقت G تكون present in a tautomeric form, the G now can match with thymine instead of cytosine. نفس الconcept here, guys. If C is present in its rare tautomeric form, بطلت بتاخد G صارت بتاخد مع A. So CA represent the rare base pairing arrangement. So رح ناخد form, guys, هون an example. We have a base pair TA. So T is present on the first DNA strand. On the second DNA strand, the complementary base is A, so TA base pairing. If T is converted in its rare tautomeric form, the T will not base pair anymore with A. Instead, the rare tautomer T now will recognize a G. نفس الكونسبت اللي شفناه هون guys. معناتها هلا بطل عنا a normal TA base pairing، صار عنا a tautomer T matched with G. If the system, the repair systems in our cells cannot repair this error, in this case, the TA, the initial form, after several rounds of replication, رح يتحول ل CG. So هون the TA guys, the TA base pair now is converted into CG, and this type of mutation is known by transition mutation because you converted a pyrimidine into pyrimidine and a purine into purine, so it's a transition mutation. In the next slide رح أوضح لكم أكثر this process ورح فرجيكم how during DNA replication you can change the base pairing and introduce an error. So بدي منكم guys تبلشوا تطلعوا معي على this DNA parental DNA strands. The first sequence, the first strand is ACGTC and the complementary strand is TGCAG. كلنا بنعرف إنه the process of DNA replication is a semi-conservative process. Semi-conservative means that the two strands will separate and each one will serve as a template for the synthesis of another DNA strand, new DNA strand. So during replication, DNA replication, لاحظتوا guys, the DNA parental strands separate and each one will serve as a template for the synthesis of a new DNA strand. Let's consider, in this case, G was converted into the rare form of G, اللي هي now can accept T and not C anymore. So G in the rare form, the tautomeric form of G, now will match with thymine instead of cytosine. So شو طلع معنا guys بالsequence after replication? ACGTC in the first strand. However, the newly synthesized DNA strand now is TGTAG instead of TGCAG. In the next round, so in the second round of replication, these two strands also will separate and each one will serve as a template for the synthesis of new DNA strands. So رح يفتحوا عن بعضهم guys. هون رح ترجع عنا a normal G. So رجع الجوانين رجع عامل a tautomeric shift. So from بلشنا أول شيء هون إذا انتبهتوا from keto to enol. هون رح يرجع from enol to keto. يعني رجع للnormal common form. يعني هلا ready to accept the normal cytosine اللي نحن بنعرف إنه G accept in a C. However, in the second case, guys, T طلع معنا after mutation. It's a normal T. ونحن بنعرف مين البيز يلي بيجي complementary to T is A. So A adenine will match with T. So شوفوا guys, after 2 rounds of replication, what will happen to the DNA? In the first DNA strand, the parental one, we have the GC base pairing. But after 2 rounds of replication, and due to the introduction of a tautomeric form of the guanine instead of the common normal form, the GC was replaced by AT, and it's considered as a transition mutation because the G was replaced by A, purine, so it's a transition mutation. In addition to the substitution, guys, تتذكروا درسنا in our previous lesson, point mutation are divided into base pair substitution and base pair insertion or deletion. So we covered the first one اللي هي كانت base pair substitution, the replacement of base pair. And now we will cover the second point is the insertion and deletion. The insertions and deletions, guys, also can occur spontaneously during replication. So guys بدي منكم تطلعوا على الفيجر وخدوا معي please this sequence in gray representing the template strand يعني the one being read by the DNA polymerase in order to produce in red the newly synthesized DNA strand. I will start by scenario 2. Let's consider guys take spontaneously the DNA template will loop out. So تخيلوا guys هون كان في عنا a stretch of thymines. عنا T شايفينهم guys هون we have 5 thymines T. In this stretch guys of thymines by chance the template will loop out طلعت لبرا one of the thymine ظهرت شوي لبرا creating like a loop at the level of the template. When this loop occurs at the level of the template, the DNA polymerase reading the template will miss this looped out thymine. ما رح تقدر تقراها. So إذا ما قريتها يعني ما رح تقدر تحط adenine at the level of the newly synthesized strand. So what will happen at the level of the newly synthesized strand? Here you will find the deletion of one base. So here it's an example of a deletion that can occur spontaneously during replication. In scenario 1, guys, the looping out now will occur in the newly synthesized, not in the template like in the first example. هالمرة by chance adenine هون example during the synthesis of the newly DNA strand DNA polymerase عم بتحط A. One of this A will loop out تظهر لبرا. So when this A looped out ظهرت لبرا so رجع عنا هون a free space the DNA polymerase will introduce a new A. So at the end what will happen in the newly synthesized DNA? In the newly synthesized DNA طلع عنا one extra A, so it's an addition at the level of the DNA newly synthesized. So additions or insertions also can occur spontaneously during replication because of the displacement or looping out of bases from either the template, so بيطلع عنا a deletion, or in that newly synthesized DNA بيطلع عنا an insertion or addition. شفنا guys how we can create a spontaneous mutation during DNA replication. But also you can create spontaneous mutations other than simply to convert the bases into a tautomer form or insertion or deletion. By chemical reactions also you can create a spontaneous mutation. رح ناخد two examples guys هون عبارة عن spontaneous chemical changes. انتبهوا guys بعدنا عم نحكي عن spontaneous mutation means naturally occurring. So نحن بنعرف in our cells different chemicals reactions can occur naturally. So مش عم نحكي عن chemicals جايبينهم من برا. Okay, so it's a spontaneous chemical changes. رح ناخد two examples. The first one is known by depurination and the second one is known by deamination. Let's start first by the depurination. من اسمها guys, depurination means the removal of a purine. So it's the loss of a purine from the DNA sequence. If we move to this figure guys, let's consider this is the DNA sequence, so one strand of the DNA with pyrimidine single ring, then purine guanine, it's a double ring, and then we have a pyrimidine single ring. Sometimes by a chemical reaction known by depurination, the bond between the guanine and the sugar will be reduced or will be destroyed. So when you cleave this bond between the guanine, it's a purine, and the sugar deoxyribose, This will create an apurinic site, means the loss of the purine, no more base at the level of this nucleotide. So in this nucleotide, you will find only the phosphate linked to the sugar, but with no base. So the apurinic site is produced when you lose one of the purine from the DNA sequence, when the bond hydrolyzes between the purine, the base purine, and the deoxyribose sugar. Why it's a depurination? Why we have more depurination more than depyrimidination? Because this bond between the purine and the sugar is less stable, is much less stable than the bond between the pyrimidine and the sugar. So that's why you can cleave more easily this bond between the purine and the base more than between the pyrimidine and the base. So that's why you can create an apurinic site. شو بيأثر هالapurinic site على البروسيس of replication؟ When the DNA polymerase read the sequence، قريت أول وحدة شافت في base، حطت بقبيلها another one، وصلت على هيدا البارت اللي هو it's an apurinic site. At this level no more base, so the DNA polymerase cannot introduce a complementary base in the newly synthesized DNA. So at this level, the DNA polymerase will dissociate from the DNA and the process of replication will end. The second type of a spontaneous chemical change is known by deamination. من اسمها deamination, it's the removal of an amino group. رح ناخد one example. It's an important example, guys. It's the cytosine, the deamination of cytosine. This is the chemical structure of cytosine. شوفوا هون at this level, at the level of carbon number 4, you can find the amino group NH2. If you remove this amino group by deamination, you will convert the cytosine into uracil. ونحن بنعرف إنه الuracil عادة it's present at the level of the RNA and not found at the level of the DNA. So let's consider, guys, this is a DNA sequence. Here at this level كان عادة عنا a cytosine, but after deamination, the cytosine was converted into uracil. So صار عنا U facing the G. If uracil was repaired by the repair systems, so uracil رجعت cytosine, so هون ما في error, and this will minimize the mutational consequences of the cytosine deamination. However, if the uracil is produced after the deamination of the cytosine is not repaired, so the uracil, نحن بنعرف at the level of the RNA match with adenine, right? So here at the level of the DNA, uracil will match with adenine. And then during another round of replication, the adenine, تعرفوا at the level of the DNA, the normal base pairing is with thymine. So شو رح يطلع عنا at the end, guys? The first base pairing was CG before deamination, and after deamination was converted into TA. So we converted the CG into TA, and it's a transition mutation, converting the pyrimidine into pyrimidine. In addition, our cytosine, guys, present in our cells, تتذكروا درسناها بchapter 18, could be methylated. ذكرت one, the CpG island at the level of the promoter, the cytosine can be methylated. So any methylation of the cytosine convert the normal cytosine into a 5-methyl cytosine. The deamination of the 5-methyl cytosine convert this base into thymine. Okay, so إذا عملنا deamination لل 5-methyl cytosine رح يتحول ليصير thymine. So خلينا نشوف what is the consequence of this 5-methyl cytosine deamination. I will start by the DNA sequence, the normal one, G complementary with 5-methyl cytosine. In the first step, I will deaminate the 5-methyl cytosine. So I will convert the 5-methyl cytosine into thymine. and during another round of DNA replication, the thymine عادة مين بياخد بوجهه adenine, so you will convert the G5-methylcytosine into AT. It's a transition mutation. خلصنا أول part, guys, related to spontaneous mutation, naturally occurring mutations. Now we will move to the second part, and we will discuss the induced mutations. Induced mutation مثل ما اتفقت أنا وياكم at the beginning of this lesson إنه induced mutation معناتها we need the presence of a mutagen, a physical mutagen or a chemical mutagen. Okay, so we need external factors. It could be a physical or chemical agent known by mutagen. We will start by the physical mutagens. Example of physical mutagens are the radiations. We are exposed to different types of radiations, guys. All forms of lives are exposed to radiation and continuously to radiations. كل الوقت نحن هلا وين ما كنتوا قاعدين وين ما رحتوا شو ما عملتوا, we are always exposed to radiations. These radiations are divided into two types. We have natural sources of radiations and we have man-made radiations. Examples of natural sources of radiations, we have the cosmic rays, the ones produced and projected into the Earth from space. We have also among the natural sources the radon. And we have the radioactivity from the decay, the breakage, the decay of rocks and soil. So the decay of rocks and soil will produce radioisotopes. The decay of radioisotopes will produce radioactivity. تخيلوا guys نحن هلا إذا قاعدين بالبيت الحيطان اللي معمرة منهم البناية ما هن من وين جايين؟ جايين من rocks and soils. So الحيطان اللي نحن قاعدين حواليهم surrounding our rooms بقلبهم radioisotopes, and the decay of these radioisotopes رح تعطينا radioactivity. Among the man-made sources, guys, we have the X-rays اللي هن بنستعملهم for medical uses. We have the cathode rays tube display, the one present in the old إذا بتتذكرون TV screens and computer monitors. بيبقوا التيفيز كتير كبار من ورا لأنه بيجي بقلبهم cathode rays. These cathode rays will produce certain type of radiation. And also you can be exposed to radiations from watches and other devices that glow in the dark. So these three examples represent the man-made sources of radiations. Radiations are divided into two forms, guys: ionizing and non-ionizing form. I will start by the ionizing radiation. من اسمه, ionizing means will produce an ion. So let's consider we have this molecule. This molecule with أكيد عم نطلع على these electrons rotating or moving around orbitals. If the ionizing radiation has enough energy to knock out, means to remove the electron out of the atomic shell, so this molecule will be converted into an ion, and this ion will affect the bonds between the nucleotides at the level of the DNA. So this ion produced by the ionizing radiation can break the phosphodiester bond between the adjacent nucleotide at the level of the DNA, leading to mutation. However, the second type, known by non-ionizing, من اسمه يعني will not produce an ion. All ionizing radiations induce mutation. However, the non-ionizing non-ionizing radiations, all types except the ultraviolet lights or UV, does not induce mutation. Okay, so all types of ionizing mutation, all types of ionizing radiation induce mutation. However, all types of non-ionizing radiation do not induce mutation except UV or ultraviolet light. So اللي بدنا نعمله guys إنه بدنا ناخد example of the UV light. UV light, guys, it's the specific type of radiation produced by the sun. وعم توصل على الأرث, guys, we have two types, UVA and UVB. أكتر شيء نحن بنكون exposed to UV وقتا نروح على البحر, بنكون كتير exposed أو وقتا نمشي بالشمس. This UV light, guys, has a certain amount of energy which increase the chemical energy of certain molecules, specifically the pyrimidines اللي هن cytosine and thymine bases in the DNA. So رح ناخد example, guys. هون we have a DNA strand. In this DNA strand we have two adjacent thymine, يعني على one strand of DNA في أول T وحدة another T. If our DNA was exposed to UV light, the UV light will increase the chemical energy of the thymines and will create or form a chemical bond between adjacent pyrimidine molecules in the same strand of the DNA double helix. So رح يطلع عنا a bond, هون a bond between the two thymine, leading to the production of a thymine dimer. هاي it's an example of thymine, بس ممكن يكون عنا cytosine or cytosine thymine or thymine cytosine. So different, إذا بدكم possibilities, different combinations of pyrimidines كلهم بيمشي حالهم, بس أكثر شيء نحن بنشوف between two adjacent thymine to create a thymine dimer. So it's an example of pyrimidine dimer. This bonding, guys, between the two thymine residues or thymine bases will create like a bulb in the structure of the DNA. So this is the structure of the normal DNA strand, guys. إذا لاحظتوا حناخد هون the two yellow bases, they are considered as the pyrimidine bases. Let's consider these two yellow represent the thymine. So we have two thymine bases adjacent to each other, and this DNA was exposed to UV light. The UV light will increase the energy of the chemical bonds between the thymine, creating a thymine dimer. This structure, guys, represent after the incoming of UV light. وشوفوا how the thymine bases form a bond, a chemical bond between them. So this chemical bond will disrupt the normal pairing of T with A. هون كان في عنا T with A. هون the thymine, when they form the normal, when they form the chemical bond, the thymine dimer, they cannot attach anymore to the adenine. So this will create a bulge in the DNA strand. And when the DNA polymerase read the DNA template during the process of the replication, the DNA polymerase cannot proceed past the lesion here, and the process of replication will end. And if the cells cannot repair this thymine dimer, the cell finally will die.